And I need my clicker. Oh my gosh. Good morning. Okay, so don't forget, you have to do the 2 ways that I'm taking attendance. I'm gonna think about a different way so that maybe we just don't do iClicker. I don't know. I'm thinking about it. But for right now, please enter your name on the spreadsheet and check in on iClicker. Still have a few minutes, but please check in, put your attendance, and then iClicker.
That's an idea. But if you have like a place on the
That's what I'm
spreadsheet
thinking for like Zoom people.
Yeah.
That's what I think I'm gonna do. I think I'm just gonna put like at the bottom, if you're on Zoom, then in attendance, put your name. Because this is what I was thinking about. That's exactly what I was thinking about. Then we can kind of eliminate.
Right, yeah.
Yeah, that's what I think I'm gonna do. I keep changing it every class period. So let's try that. Let's try that. Those of you who are on Zoom, scroll down. I am on Zoom. So put your names here. I'm gonna, but I don't want people to type over each other. So just make sure that when you do this, if you're on Zoom and Zoom only, type your name down here. We'll put a couple of spots. If you are only on Zoom, go ahead and check in. We'll put just a bunch of spots down here. And then maybe we can get rid of the other attendants. Hope that works. Oh, and I'm not monitoring my chat. Yay. Okay, thank you, Sam. I see you did. You're awesome. Right. If anybody else is on Zoom, go ahead and put your name at the bottom in these yellow squares. Don't forget to put your name, and don't forget to click enter or click a white box after you finish typing. Good morning, how are you? Anywhere there's an open one? Okay. All right, y'all ready? Good one today. It's a really good one today. I say this every day. Why was the broom running late? It overswept. It's a good one. That's a good one, right? I thought that was appropriate for Monday morning. I felt like that this morning myself. So if you have not done so already, please type in your name on the Excel spreadsheet. If you are joining us just on Zoom, type it in down here in one of these. All right. Are we gonna rock it out on a Monday morning? Okay. Please also make sure just for today that you also check in on iClicker. I think starting on Wednesday, I think we can nix iClicker if we have a place for people who are on Zoom to check in down here. So if anybody has any issues with that, let me know, but that's kind of my plan. Okay, and everybody knows where to go to get the lecture slides and everything that I do in class, right? Right here, slides of all chapter PowerPoints. This is what I'm going over in class. Okay, so, oh, and there was a issue with chapter 1 homework and reading quiz. So our class is in a different time zone than the publisher. So it ended an hour early for everybody. So what I did was I went ahead and extended it until tonight at midnight and then had them reset the time zone for our class. So it was a time zone issue if that happened to you. But it should be fixed from here on out. If anybody has any issues like that again, please let me know, but it should be fixed. Okay. So let's start chapter 2. Are you ready for chapter 2? And I am going to look, I did not get to look at all the discussion over the weekend, so I'm going to look at it this afternoon and post comments on the chapter 1 discussion so that y'all have that. But it was looking pretty good. I checked it on Friday when people were still kind of working on it. It was looking really good. Okay, so chapter 2 is all about physical biochemistry. What do we mean when we say physical biochemistry? So physical biochemistry is all about energy. So energy conversion. There's a particular name for energy conversion in biological systems. It's called bio, of course it's bio, energetics. E-N-E-R-G-E-T-I-C-S, bioenergetics, I think. So we'll talk a ton about section 1. Section 1's huge in this one. Then we'll talk specifically about water, about why it is so insanely important and so insanely cool. So let me ask you this. How is an individual water molecule held together? One water molecule. Water. H2O. Each of these is a covalent bond, right? But what holds water molecules to water molecules? Hydrogen bonds. So should I draw a line between my oxygen and my hydrogen? Why not? It's not a covalent bond. So whoever named hydrogen bonding, I could slap them. You all know I'm nice. So that's saying a lot. But like maybe not slap them, maybe just shake them a little bit. Slaps kind of, yeah, shouldn't do that. Maybe just a little shake, right? Okay, so what is a hydrogen bond? A lot of times you'll see it denoted as like dots. So that is a hydrogen bond. So what is a hydrogen bond? If it's not a bond, not a covalent bond, right? What is it? So each water molecule is polar, right? That means it has partial charges. Y'all remember partial charges? Y'all remember how to denote partial charges? Multiple ways? Yeah. So a sigma plus and a minus because the oxygen hogs all of those electrons. Remember that? The other way that you could do it is what? You don't have to do both, but I'm saying either one. Is you draw the little arrow that has the plus. Remember that? So you do both. Either way, it's the same thing. So then why does this oxygen interact with the hydrogen in the hydrogen bond? Because we have a partial negative on the oxygen, a partial positive on the hydrogen. And so that gives us an electrostatic interaction, right? A partial positive and a partial negative being attracted to one another. An electrostatic interaction. It is not a bond. Not a bond. I don't know how many times I've said that in my teaching career, but I feel it coming on for many years to come. All right, so the interactions between water molecules are non-covalent, but they're insanely important, right? I just downgraded a hydrogen bond, right? But at the same time, I'm telling you that they are insanely important because lots of small interactions can make a big impact on things at the molecular level, right? Like DNA base stacking interactions are super insanely important, but they're minuscule, right? But we have tons of them. Same thing with hydrogen bonds in water and in all other kinds of hydrogen bonds, right? And so we'll talk about how all of this, why water is important in terms of things like pH, in terms of pKa, in terms of buffers, all that kind of stuff. And then in lab, you're going to experience that if you're taking the lab as well, right? And then at the end, we'll talk about cell membranes and how they function. But that's like a tiny, tiny section, right? So let's talk about energy conversion, because energy conversion is what drives everything, right? So we know that there are producers and there are consumers, right? Back to biology 155. So our producers do what? They produce chemical substances, right? And how do they do that? Where do they produce it from? The sun, light energy, right? The sun. Now, are we producers? No. So we can't do that. So what do? We consume. We consume things that can do photosynthesis, or we consume things that already consumed things that did photosynthesis, right? Okay, so sunlight is our primary source of energy on Earth. It's not the only one, but it's the major one, right? And so either you have something that's going to transform solar energy into chemical energy, or you have a process where you convert chemical energy from one substance to another substance, right? Those are basically the only two things that we're going to do. And so plants in the daytime convert solar energy to chemical energy. What do they do at night? Right. They do what we do. At night, right? They can't do photosynthesis. So how do they make energy? They break down the sugars that they made during the day. So I think everybody comes in with a misconception about plants, right? You know, plants produce sugars. We eat the sugars and make energy, right? Well, plants do the same thing, but only at night. So you got to kind of keep that in mind, right? So in order to understand all of the interconversions of energy in all these living systems, we have to go back to the basics. We have to go to the laws of thermodynamics. So we're going to talk a lot about the first and second laws today. And then in order to make metabolism go, metabolism is all of the chemical reactions that happen in a living biological system, right? We have to have both exergonic and endergonic reactions. Remind me, what are exergonic versus endergonic? You did this before, right? You've seen exergonic and endergonic. Yes? Yes, okay. So exergonic, tell me about mathematically, an exergonic reaction would have a, what? Yes, yes. So energy would be released to what? To the surroundings. So what would we mathematically denote that as? A negative delta G. Yep. So typically these are going to be spontaneous reactions, right? And if it's a spontaneous reaction, typically what's going to happen is we're going to yield simpler components after the reaction's over, right? Things that have low energy inside of their bonds, things like that, right? Okay, what about endergonic? Energy is consumed, absorbed from what? From surroundings. I just put surroundings. So how do we usually denote that? Positive delta G. So what is delta G? Gibbs free energy. Awesome. Okay. So a lot of times what you'll see in biochemistry is things that are positive delta G. This is when we're making things, right? We're absorbing energy from their surroundings in order to make really big proteins, in order to, I don't know, transport molecules across the cell membrane or whatever it is, right? So we just said that this is bioenergetics. This is our energy conversion inside of biological systems. And so we talked about photosynthesis, right? That's going to transform solar energy to chemical energy. And hopefully we'll end this semester on photosynthesis. We shall see. But it's also, and I don't like how they did this, but it's also, right? It's also chemical energy being interconverted. And how do we interconvert? Through redox reactions. When we say redox, redox is a shorthand for oxidation and reduction, right? So let's see how much you remember about oxidation and reduction. Oxidation, reduction. What's our little phrase? Oxidation is what? Loss of what? Of electrons. Reduction is gain of electrons. But a lot of times in biological systems, it's hard to track those electrons. So then what else do we look for in chemical reactions? Gain and loss of what? Hydrogen and oxygen. Hydrogen, both hydrogen and oxygen. So oxidation is a loss of electrons and a loss of what else? Hydrogens, and therefore it must be a gain of oxygen. So then reduction is gain of electrons, loss of oxygen, gain of what? Hydrogen. Okay. But y'all knew that, right? Just a reminder. Okay. All right. So why do we need chemical energy? What's the point? Well, we all know, like when you took physics, right? Energy is used to perform work. But work has a different definition in a biological system than it does in, for example, a physics system, right? But we know energy is essential for our survival. And there are a couple of different ways that we use energy, and we'll talk about what those are. But the example that we've talked about so far is plants, right? They're going to use sunlight energy to convert carbon dioxide from the environment and make carbohydrates. Why do they do it? They don't do it to feed us. They do it to feed themselves at night, right? They're not around just to support us. Most things are pretty generally evolutionarily selfish. And so they're really doing it for themselves, not for us. We just take advantage of it, right? And eat them. And salad's delicious, right? Okay. So what kinds of work do we have? There are three kinds that are essential for living biological systems. There's osmotic, there's chemical, and there's mechanical. And so I want to look at examples of each of the three, right? So here's our three types of energy, and the definitions on the previous slide are the same definitions that are right here, right? We're going to take solar energy, but I want to step back. How did we get solar energy? I know this one. What happens in the sun? Fusion reaction. So we take a simple element and we turn it into something more complex, right? Fusion. And what happens in that fusion reaction is you give off EM radiation. That is what solar energy is, EM radiation. So now that we get to the planet Earth, now we can have plants who undergo photosynthesis that allow us to convert that solar energy into chemical energy. So why do we need the chemical energy? And I think most people think about work in terms of biological systems as mechanical work. So mechanical work is muscle contraction. You want to go from point A to point B, you'd like to inflate your lungs, whatever it is, that's mechanical work. I think lots of people think about that one, right? But when we talk about living systems, we need to do other kinds of work. We need to do chemical work. And when we think about chemical work, right, every time you have a hamburger for lunch, what do you have to do to that hamburger? You have to chew it. That's mechanical work. You swallow it, right? What happens? You're going to chemically break it down into substances that your body can use to make macromolecules that you need, right? So it's more than just a physical chewing of your hamburger. It's a chemical one as well. So that's the degradation of our biomolecules. But now that we've degraded those biomolecules, now we're going to turn that hamburger into something else, right? We need to form different proteins within the cell. So the protein components that you ate in a hamburger are going to turn into, I don't know, actin and myosin. Who knows, right? Some kind of protein within the cell. The other form of chemical work is osmotic work. So how do molecules move across membranes, porous membranes, generally? From areas of high concentration to low concentration, right? That is passive diffusion. That doesn't require energy. But if that was the only kind of transport that you are able to do, you would not survive. We have to be able to transport things up a gradient from an area of low concentration to an area of high concentration. To do that, you need to input energy. What kind of energy are we inputting here? ATP energy. How do we get energy from ATP? I'm going to tell you this is a trick question. What happens in order to break any bond in any chemical substance? You break a bond, are you releasing energy? Or are you gaining energy? Really? Bonds have stored energy inside of them. So in order to break a bond, you have to put energy in and snap that bond, right? So hydrolyzing ATP does not release energy. We say that all the time in biology, don't we? But it's really wrong. And I'm going to tell you today why that is, right? It's easy to think about it that way, but it's wrong. It's inherently wrong. And we're going to talk today about that extensively. So I just want to put that out there that that's a misconception, and we're going to address it today. Okay, so now let's talk about homeostasis and equilibrium, right? All of this work that we've done is to maintain homeostasis within our bodies, right? This is maintaining our temperature, right? Maintaining concentration of ions in certain places, whatever it is, right? And it requires an input of energy. And it prevents us from equilibrating with our environment, right? So what happens if we equilibrate with our environment? We're done. We are done. We are dead, right? So that means all of our biochemical processes are no longer occurring because we are now at equilibrium with the environment. So we're no longer maintaining this internal temperature, these internal concentrations of things. We're just going to equilibrate with the surroundings, right? And so those are non-living. systems that that will do that, right? So it's the fundamental reason why all living systems need that energy conversion, is to maintain homeostasis and prevent equilibrium with our surroundings, right? So that's our fundamental reason. Why do we care about bioenergetics of systems? All right, so how do we do it? Well, we're not photosynthetic, right? So we're not going to study photosynthesis until the end of the semester, hopefully. We really do it with redox reactions. So this is really the chemical work that we do inside of ourselves. So when we talk about redox reactions and we say it's linked reactions, what are we actually doing? We're actually just shuffling electrons. We're moving electrons from one substance to another substance. But why? Why do electrons move? How do we make that happen? So how do we do this transfer? How do they move? Why do they move? You have to study redox reactions, right? Oh, wait, what does Sam say? I see. So it's electrochemical. It's all about electrochemical. It's electrochemical potentials due to differences in electrochemical potentials. So it's always going to move from something with a high electrochemical potential to something with a low electrochemical potential. That's why it's sequential, right? So what do you see in our little diagram here at the bottom, right? We have this molecule. This molecule is carrying an electron. When the molecule is carrying the electron, we say that this molecule is in the reduced state, right? Because reduction is gain of electrons. So my reduced molecule has the electron. Now, this molecule, right, is going to steal away this electron so that the original molecule converts from reduced to oxidized. And now we had our oxidized molecule, it picked up the electron, and now we have a reduced molecule. And in that, okay, and in that we are going to produce chemical work. And we're going to talk today about what exactly that chemical work is, right? And there are many, many different ways that the cells actually do this in the body. So remember, we talked about this. What is reduction? What is oxidation? We did this. So this is an example of oxidation reduction reactions that occur in photosynthesis. And in photosynthesis, when we transfer those electrons, right, the electrons that get shuffled around in photosynthesis actually come from water, right? So this is one of the reasons why you have to water your plants, right? They need sunlight and they need water, because those electrons that are excited when light comes in, right, and they get transferred from your chlorophyll all the way down the line, right? Well, how do you replace the electrons in that chlorophyll? If the electrons moved, how do you excite that chlorophyll molecule again? Well, you have to give it some electrons. So where those electrons come from, they come from water, right? And so a lot of times what you're going to see in biological molecules is that you will have what are called electron carrying molecules, right? So this happens in us as well when we go through cellular respiration, right? So if we want to perform work, let's not the plant, let's talk about us. What are we going to consume typically? The most common thing, glucose, right? So what's the formula for glucose? H12O6, right? Okay, so in cellular respiration, what do we produce? What are we going to get out of this? Why do we eat things that have sugar in them? Y'all, it's really a Monday, huh? What do we make? We make energy, we make ATP. So we can say energy. That's true. What else? CO2. What else? Water. Okay, good. All right. So what's happening to the sugar? What do we see? A loss of what? Am I seeing electrons in this? No, I can't really see electrons. Well, what can I see? A loss of something. We're losing hydrogens, right? We made a carbon dioxide molecule. Do we have any hydrogens in there? No. So what happened to our carbon-containing molecule? It lost those hydrogens. If you lose hydrogens, you decrease in the number of hydrogens, what's happening? Oxidation is happening, right? But at the same time that oxidation is happening, where are those hydrogens going? They're going to electron carriers. And in biological systems, those electron carriers tend to be NAD plus, and NAD plus gets converted into, anybody know? NADH. How about FAD? What do we turn FAD into? FADH2. H2. Notice that NAD plus had a plus on it, right? FAD did not. But those are examples of electron carriers. And so as we move those electrons along those electron carriers, we can actually do work, right? That's how it happens. Okay, so we have to kind of back up just a little bit and really talk about our thermodynamic laws. So before we talk about thermodynamic laws, we need to know that we're all on the same page on vocabulary, right? So if we're talking thermodynamics, our system is any collection of matter in a defined space. That's pretty broad, right? So that could be a test tube, right? I'm going to pick that it's a test tube. What else could it be? The whole body, one organism, could be one cell, right? It could be a lot of different things. Anything we choose to study. The system is our choosing. The surroundings are all the space that's not included in the system, right? So everything around that system. So what happens when we add the system plus the surroundings? What do we get? The universe. Universe. Okay. So I feel like this is like physics, right? In physics, there are multiple types of systems. And as you read this, I want you to think about what's a biological system. Which one of these three is a biological system? So you can have open systems where both matter and energy are exchanged with our surroundings. You can have closed. Closed is only energy being exchanged. And then isolated is nothing is exchanged. So which one is a biological system? Open. A biological system. Good. So what is the matter that is exchanged for a biological system? Nutrients. Nutrients can come in. What goes out? Waste. Okay. And then energy. Energy is exchanged. How do we exchange energy? You're exchanging energy with your environment right now, with your surroundings. Heat. Heat. Okay. So let's talk about those laws of thermodynamics. So our first law of thermodynamics, we're really going to talk about the first and second. Those are the two big ones that are really, really important for biological systems. So our first law of thermodynamics tells us energy cannot be created nor destroyed, right? It can only be converted. So when we look at energy of a system, typically in systems, we're going to look at changes in all these parameters that have to do with thermodynamics. So we're not just going to look at E, we're going to look at delta E, right? Because delta is the difference in final versus initial, right? And like everything. Okay. So that change in final and initial energy is really dependent upon two things. What two things? Q and W. What's Q? What's Q? Heat. Q is heat. What is W? Work. Okay. Well, what is work? This is physics flashback, right? It happens. It's okay. So what is work? What? How do we find work? These are changes in what? What two parameters typically? Pressure and volume. Let's talk to me about a biological system. Can I change the pressure of a biological system? My cell will explode or my cell will shrivel, right? I can't do that. I can't change pressure. Can I change volume? Not really. Yes, there are some really, really, really small volume changes, but if your volume changes too much, think about a plant, right? If you don't water your plant enough, right? What happens to your plant? Shrivels, right? They get really sad. Like you can come and look at the plants in my office. Periodically, they look really sad because I'm terrible about watering my plants. But you really can't change volume without having a detrimental effect on the cells, right? So we really can't change those two. So a living cell has to convert energy in constant pressure and constant volume, right? So to measure the energy of a system, we need to talk about another parameter. We need to talk about enthalpy. So enthalpy is H, right? So energy has an effect on the heat content. And this is one of the things that we said we do exchange with our surroundings. So obviously it is going to be really important in factoring in what we can actually do in energy conversions in the cell. So to measure our energy change in a system, right, we have to talk about enthalpy. So when we talk about enthalpy of a system, remember H is equal to E PV, but we're not gonna use that straight formula because in biological systems, we can't measure absolutes. It's really, really, really difficult. So we typically measure changes in. That's why you're going to see them all as deltas, right? Okay, so that's our first law of thermodynamics. This is the one that's going to be really important. Oh, and talking about that, talking about formulas and talking about important formulas. If you have a hard copy of the textbook, I don't know how, I'm going to ask y'all with the online textbook. It's either front or back. I don't remember offhand now. I think it's in the front. There's a sheet of equations. I'm going to give you that sheet on the exam. Those of you who are online, would y'all look and see if that's provided to you in the online? Has anybody seen it offhand? Go and look and somebody let me know, hey, I found it. You know, if not, I'll post it on Moodle so that y'all can see exactly what you'll have on there. I think I did post it on Moodle. I actually think I already did. But anyway, I'll give you the list of formulas. Okay, so when we talk about enthalpy, right, there are two different kinds of enthalpy. Either things are exothermic or things are endothermic. So exothermic tells us that things are exiting, things are leaving, right? So it releases heat. And so we have a negative value for delta H, right? If it is endothermic, it's pulling heat in, right? And so it is absorbing heat, and that's why we have a positive H, right? Remember this? Okay, it's gonna be really important that we keep these straight in the future. Okay, so the second law of thermodynamics. So you have to remember that everything in the universe tends towards disorder, right? If you have a pack of cards, how many different ways can you organize them? 52 cards. A ton of different ways, right? A ton. So if you wanted to put them right in you know, like hearts, diamonds, spades, clubs, and you wanted to put them high to low, right? You could do that. But then if you shuffle, how many different combinations are you going to get after that shuffle? How likely are you to get that original one back? It's not, right? It's not going to happen. So there's so much disorder, and the universe tends towards that disorder, right? In the absence of an energy input, I could pick up those cards and I could sort them, right? But that's an energy input that I'm doing. I'm actually putting energy in to make it more organized. So we're always looking at increasing entropy. And when we look at entropy, right, it's the universe, not necessarily of a system or the surroundings, but of the universe itself. Because we do have some things that are spontaneous, right? And so we're going to explain how that happens. So you have to remember one thing about all biological systems. Biological systems are not theoretical, right? So you're going to have some energy that is lost as heat. So in biological systems, some energy is lost as heat. So when we do some calculations, right, and then you compare it to actual values of energy output, it's not going to be perfect. And that's because we're always going to lose a little bit of energy to heat, right? Okay. So if we have biological conditions where we have no change in pressure, we have no change in volume, right? We can combine equations in terms of energy and enthalpy, right? So my change in enthalpy is equal to my change in, my change in energy is equal to my change in enthalpy minus, we know, pressure times volume, and that is equal to Q minus W, right? That's from before. But what did we say about a biological system? What is pressure and volume? Constant. So we're not going to change it. So change is 0. How about work? Work was what? Depending on pressure and volume. So work's going to be what? 0. So if we rewrite our equation, what do we get? Delta E is equal to delta H, which is equal to Q. But knowing delta H doesn't necessarily tell us if the reaction is favorable or not. We need Gibbs free energy for that, right? Because y'all told me just a few minutes ago, I asked you what would happen to a spontaneous reaction, right? And you gave me values for delta G. So delta G is what we really need to calculate. Do we have everything we need to calculate delta G? We can't just have enthalpy. What do we also need to have? Entropy. So we need to be able to combine enthalpy and entropy. So how do we do that? Well, we have to talk about entropy a little bit, right? So when we talk about chemistry, really entropy a lot of times is mostly energy due to motion. So in chemistry, usually it's energy due to motion. So if it's a measure of disorder, the more motion we have, the more entropy something has, right? So if you look at a large biomolecule, there's not a whole lot of movement that can occur with the individual components of one really big biomolecule. It's basically stuck in its three-dimensional shape and can't move too much, right? But what if I take this one big biomolecule and I break it down into a lot of little ones? Now tell me about the motion. Now they're free to move. You're free to have more resonance in bonds. You're free to have just general molecule rotations, right? So as we go from a large molecule into small molecules, what happens to delta S? So if we have one big molecule and we convert it into a bunch of tiny, what happens to delta S? Delta S increases. That's the same thing when molecules move in a gradient, right? From areas of high concentration to low concentration. As they move to those areas of lower concentration, they have more freedom of movement. So the same thing happens. Okay, so let's look at water real quick because we talked so nicely about water. We already know about hydrogen bonds and everything, right? So when we're in a solid phase, water is in a crystalline structure. If you have a crystal of anything, those bonds are not changing. They're fixed, right? And in water, what do you see inside of that crystal? In these places where my cursor is going, I see really big what? Open spaces, really big open spaces. So when water's in a crystalline form, because those hydrogen bonds are basically fixed, they can't be changed, then it's going to have a lower density because the molecules are more spaced out, right? And it has a low amount of entropy because they're fixed. They can't really move. They can kind of shake, but they don't move, right? There's some vibrational energy, but not a whole lot, right? But what happens if I take an ice cube out of my freezer and put it on the counter? What's going to happen to that ice cube? It's going to spontaneously melt, right? Why is it going to spontaneously melt? The universe tends towards disorder. So tell me about my entropy from going from ice to water. Now, because I'm in liquid water form, hydrogen bonds form and break, form and break, form and break, form and break, form and break. And so all those water molecules are kind of jumbled together. That's why the density is greater in water, liquid water, than it is in ice, right? It's magical that your ice floats. No other substance on Earth does the solid float in the liquid. It's bizarre. But we see it every day, so we think it's normal. But it's not, it's bizarre. So in liquid water, those bonds are forming and breaking all the time. And so does the three-dimensional shape of liquid water have a three-dimensional shape? Does it even have one? No, right? It's all constantly changing. That's why it's in the liquid phase. So you have those fluctuating hydrogen bonds. That's what leads to our higher entropy. Okay, so that's part of it, right? But our spontaneity also depends on temperature, right? So what if I took that liquid water and I put it in a beaker on a Bunsen burner and I lit the Bunsen burner? What would happen? I would get gas, right? Okay, so tell me about gas molecules. We don't have a whole lot of hydrogen bonding going on in gas, right? Those molecules are too far apart. They can't actually interact. They can't make those hydrogen bonds. Okay, so what does that tell me about entropy? If I increase temperature, what happens to entropy? I increase entropy. Okay, good. So my entropy is dependent upon two major things. What two major things are my entropy dependent upon? Temperature. So S depends on temperature. And what else? And what controls motion? That's right. Motion is right. What controls motion? Intermolecular forces and intramolecular forces. So we'll just say number and type of bonds. And we'll kind of do that. Right? Okay. So now we've talked about enthalpy, we've talked about entropy. Now we have to talk about Gibbs free energy. So to determine if any given chemical reaction is favorable or not, we have to figure out Gibbs free energy. Now, there are different kinds of Gibbs free energy. There's just plain old Gibbs free energy, right? It's not equals. I don't know why I did that. It's not equals. So there's Gibbs free energy. Then there's the change in Gibbs free energy, right? But if you're reading in the book, you notice that you have delta G naught. What is delta G naught? They read? You should be reading before you come to class. It's standard free energy. Standard. So there are particular conditions that must be met in order to have standard free energy. One of them being concentration of a substrate being 1 molar. It's not the only one. You have to have constant pressure, 1 ATM. And I'm just going to go ... because we're going to talk about what those are in a minute. And you'll have them on the slides, so don't freak out. Then you have standard Gibbs free energy of a biological system. So this is standard of a biological system. In every chemical system, are we going to have things that are at a concentration of 1 molar? That's bizarrely crazy. No, we're not, right? Concentrations of substrates change all the time. So something can have a mathematical standard Gibbs free energy that makes it non-spontaneous. But when we put it into the biological system, those concentrations change, and they change, and then maybe that reaction becomes favorable. So it's important to understand that there is a difference between standard free energy and biological standard free energy, a huge difference. And I feel like that's something y'all should know, like on the front end. Okay, so if we want to calculate free energy, we can't go in and calculate Gibbs free energy. We have to calculate change in free energy. So if we want to do that, whoops, what we need to do is convert that formula to looking at changes from one condition to another condition, because that we can measure. But it's really hard for us to measure standard free energy in a biological system. So we look at change in. So y'all know, some people in here do know what I call this formula. This is the ghetto homies formula. So I make up these really funny ways to remember formulas. And so every time I write it, that's what I think in my brain. I know it's kind of crazy, but like, I'm going to share those things. That's like cars in the garage and apples in the tree. I share those things with you because it helped me remember. So I'm hoping it helps you remember too, right? Okay. So when we look at this equation at equilibrium, delta G is equal to 0. Chemically, what does that tell us? What's the definition of equilibrium? Right, we have A going to B and B going to A, right? So we have a reversible reaction. what does that tell me about this reaction? The rate going forward is equal to the rate going back, right? It's not that the reaction isn't occurring. It's that the rate of the forward and the reverse reaction are equal. So the formation, the rate of formation of products and reactants is equal. It's the same thing that I just said, right? Rate of forward is equal to rate of reverse. Same thing. We just said it different ways. Okay. So now we come back to what we were talking about in the very beginning, right? If something is endergonic, what's our delta G value? It's going to be negative. If something is endergonic, positive. So if we think about endergonic and exergonic in terms of those reactions, if something is exergonic, it's going to be seen in our forward reaction. We're going to favor the forward reaction, right? The rate of the forward reaction is going to be greater than the rate of the reverse reaction. Okay, that's a favorable reaction, and that's going to be spontaneous. Endergonic is going the other way, right? We're going to favor the reverse. So the forward reaction is going to be therefore unfavorable and non-spontaneous. Yeah. So like all my biology people are like, the beginning of biochem sucks because it's so much chemistry. And my biology people and my chemistry people are like, yes, I know all this, you know, but trust me, whatever position you're in, it's going to flip flop throughout the semester. So don't freak out. All right. How to tell if a reaction is spontaneous or not. You could do one of two things. You could memorize this table. I hate memorizing tables. I hate it. I think it's disgusting. I hate memorization like that. So I'm going to teach you a different way. How about that? I like that. How much time we got? 5 minutes. We can do this. We can do this. What? Just no. We end in 2 minutes. How long we got? 2 minutes. 2, 3. Okay. All right. So what's our formula? If we don't finish, it's okay. We'll pick up next time. Delta G, Gibbs free energy, ghetto homies. Yes, come on, y'all. Okay. So in order to be spontaneous, what do we need? We need a negative delta G, right? You all agree with that? Okay. What does delta G depend upon? Look at your formula. Tell me what values it depends upon. Delta H, right? So there are two cases that could happen for delta H, right? Delta H could be negative. What is that called? Exothermic. Delta H could be positive, which is endothermic, right? Okay, what else is it dependent upon? Extend page. Look at your formula. Give me my other one. Delta S, right? What are my two possibilities? I could have a negative delta S. I could have a positive delta S. A negative delta S tells me low entropy, right? Positive delta S tells us high entropy. Okay. What else are we dependent upon? Temperature, like the absolute value of the temperature, right? Okay, there are 4 different things that could happen. So I'm going to extend my page even more. There are 4 things that could happen, right? I could have something that is exothermic, but have a high amount of entropy, right? I could also have the opposite of that. I could have something exothermic, but with low entropy. Yes? What are my other two possibilities? Endothermic with high delta S, or I could have endothermic with low delta S, right? Okay, so when we come back next time, what we're going to do is we're going to take this, we're going to fill in our formulas, and we're going to predict what's going to happen, if something's going to be spontaneous or not. Because it's way better than memorizing a table. All right. Y'all are free to go. Anybody on Zoom need anything? Okay. So I'll go ahead and end the Zoom. Have a good rest of your Monday.